everybody. In this podcast, our third in our energy domain, we are going to spend some time with enzymes and how cells control their metabolism and how living systems carry out these specific chemical reactions, because as we know, they're a pretty important key to all functions of life. So let's groove it. Okay, so lots of stuff going on here with enzymes. They're the big ones. So enzymes are what we're going to be looking at primarily here. They are our biological catalysts. They help to speed up reactions and allow them to occur at much lower activation energy. So it takes and requires less free energy to make the enzyme run it, or excuse me, to make the reaction run its course and still get it in the time frame that it needs to be in. So without an enzyme, the course of the reaction need really, really high free energy, and with an enzyme, it doesn't, so which is nice. And the actual change in G, so that entropy piece is really unaffected by the enzyme when we get our products. So the actual end product piece isn't really as effective. For the most part, our enzymes are proteins, and some RNA molecules act as enzymes. So we'll maybe be exploring potentially what some of those are. You're going to find that most enzymes end with ASE. So ACE, you're going to see that a lot coming up as we talk about enzymes. So like catalase, like what we used in the lab. So just something to remember. It's common nomenclature, and it usually is based off of whatever reaction the enzyme or the substrate that the enzyme catalyzes. So just to kind of keep that in mind. So how do enzymes work? We call this the induced fit model. Enzymes interact with the reactants, which we will refer to here on out as the substrate. They actually will bring the substrates together, or in fact break a single substrate apart. So they are necessary for both synthesis and decomposition reactions, and they will allow for these bonds to form where it's a more energy favorable situation. And what happens is there's a very specific area on this enzyme known as an active site, and that active site works to pull the substrate in, and it's very, very substrate specific. And for the majority of enzymes, they fit a particular substrate, and that's what they work on. So when a reactant comes in, we actually refer to that reactant as the substrate enzyme complex. There's a shape shift that happens with the enzyme, kind of puts pressure either on bonds and breaks them, or forces a collision between the two. The product then pop out, and the enzyme is back to normal. So enzymes can be used and reused pretty often. Okay. So again, like I mentioned, this is the induced fit idea. So the shape of that active site is very specific to a particular substrate. Oftentimes the enzyme is huge molecularly comparatively to the substrate that it works on, and the binding of the enzyme to the substrate induces a change in the enzyme shape to elicit the response and catalyze the reaction. So pretty small area. So if we look at this sort of space-filling model, you have the substrate and you have the enzyme in the blue, and that active site where the substrate fits right in nice and snug. So here are some examples. We have topoisomerase. We've mentioned this one before. This is involved in minimizing stress on DNA during replication to kind of keep it from wearing out, if you will, too soon. So this will make a temporary sever, I guess, the helix slightly. So this is one that's kind of an example of an enzyme that happens. Then you have rubisco. This is an important enzyme, super important in the Calvin cycle. That is part of photosynthesis, which is where we're going next. And it attaches carbon dioxide to the precursor molecules to help build a 6-carbon intermediate that is part of the steps in creating a sugar molecule. So those are just some examples of what we have. So there's a little closer look at topoisomerase, what it does. You can see the blue is actually 2 polypeptide chains. There's a quaternary protein structure here. The orange is a DNA double helix there, and the purple area would be the active site that the DNA fits into. So there's that. Now, some enzymes need a little bit of help. So the helper needs some help, and they require what we call cofactors. These cofactors are often vitamins or minerals, hence why we have to take our vitamins and our minerals all the time, actually promote and help the enzyme have a greater affinity for the substrate by binding to it, and they will actually bind so that it can create a functionality in that active site and make them work better, ultimately. So you have magnesium ions, are important for rubiscu's active site. There's also manganese ions for the topoisomerase. So these are just, again, just some metal ions and why we have other atoms, and other elements in us besides, the big six. So evolutionary considerations here when it comes to enzymes. They are, the idea of evolution plays a huge role in the structure and function of enzymes, being that they're mostly proteins. We know that their structure does greatly influence what they do. And there's a lot of look now into the evolution of enzymes. So there's things like the analysis of enzyme genes, looking at comparative sequences and how enzymes vary. Artificial selection of enzyme activity in lab sections. So like if you go, a little throwback to sophomore year when we did the spit lab. Okay, so remembering that, and so your early biology days, we can artificially select, and we can kind of create hypothetical situations where that enzyme would be more important in certain areas than not. They're actually looking at ethnographic and demographic studies of culture and race backgrounds and enzyme genotypes. And so you get the variation, you get the natural selection, and then there's adaptation in the population with regard to these enzymes. So that is just some evolutionary considerations to make. Now enzymes are regulated and they can be turned on and off pretty easily through either inhibition or activation areas. You've heard this before as called allosteric regulation. So enzymatic functions can be, ultimately enhanced, activated, stimulated, or inhibited, stopped by different factors in the cell. We have competitive interactions here. A molecule other than the substrate binds to the active site. Obviously, if there's something stuck in there, right, the substrate can't bind, so makes sense, right? And then there's non-competitive interactions where the regulation is accomplished without actually getting into the active site. It would bind to another area, causing a shift to the actual active site shape. And here's where that allosteric interaction comes in, what we call the other site. So stimulation or the inhibition of the enzyme, it causes an actual conformational change. It actually changes what's going on. Okay, so here's where we see the difference between activation and isolation. Activation, we're going to cause the enzyme to become more susceptible to binding to the substrate. In inhibition, we're actually going to shut it down. And we go back and forth between these. So a binding of an activator can stabilize the enzyme into active. Binding of an inhibitor, exactly, it's going to make it inactive. Pretty straightforward stuff, eh? Now, if the binding of a substrate on the active site also contributes stabilization of the conformation in all the subunits, this is what we call cooperativity. So, hey, better together, right? So here's an example. Capsase, important enzyme that deals with apoptosis and necrosis, so cell death. There's an experiment to determine if capsase enzymes can have an allosteric site or not. The active and inactive forms of capsase were already known, so the hypothesis was allosteric inhibition of capsase 1 locks the enzymes in an inactive conformation. So as we look here, here's the experiment, right? There's the active site, there's the substrate, we activate the form that works can fit it. Now, allosteric binding site, we have the inhibitor, hypothesis, allosteric inhibition locks it. Okay, so far so good. Well, let's look at what the results were. So there's capsase 1, there's its active form, you have allosterically inhibited form, and its inactive form. So it turns out organization. All right, so how are enzymes organized? Compartmentalization is actually a way to have enzymes separated. So you have localizations, very specific enzymes and reactions that go with them. You find only those enzymes in those areas. So it's not like these enzymes are spread all over the cell, and we will actually localize their function in various areas of the cell to allow them to be more more readily available for where those reactions need to happen. So, where that might be needed. So for example, enzymes associated with cell respiration in the mitochondria are going to be found inside the mitochondria. They're not going to be found spread all over the place. That's where they're going to be. So those enzymes necessary for respiration are going to be found there and embedded all through in various locations. Okay. There's also environmental influence on enzymes, as we have viewed and looked at in the lab. So different factors will influence the function of an enzyme. They're very specific, and not only that, these little things are very sensitive. They're sensitive to temperature, they're sensitive to protein. This is just like, sensitive to protein, sensitive to pH. Because they're proteins, and proteins we know are directly affected by this. So, change those environmental conditions can actually denature these enzymes and make them non-functional. Okay, so here we have it. So big deal of what's going to influence the productivity of an enzyme: temperature, pH, and the concentration. If we notice, as reactions are going up and continuing, right, those reactions are going to go up. They are going to slow, and they're going to slow because as more and more of the substrate is taken care of, there is less substrate for the enzyme to bind to. So over time, you're going to reach a kind of a plateau as to how the enzyme works, and it's actually going to level off really and then have zero rate. There'll be no rate of enzyme reaction because the enzymes, the substrate's all gone. Okay, yeah, that's really it. Okay, and then there's feedback mechanisms that can work. So there's a lot of metabolites in us that have regulatory effects on enzymes, and so the actual product of the reaction acts back at the enzyme and the initial enzymes itself to prevent the metabolic pathway from happening. So here's what we have in terms of feedback inhibition. So we have our initial substrate, which is threonine, goes through a series metabolic pathway of intermediates with various enzymes. The end product is the amino acid isoleucine. As the isoleucine becomes more and more and more and more concentrated, that product then acts back, okay, at enzyme 1 and actually acts as an allosteric inhibitor and changes the active site, and it can no longer catalyze threonine into intermediate A. So that means that the entire metabolic pathway shuts down and you don't have that, and it will actually slow and cease the production of isoleucine. So then it's all regulatory, right? So then as the isoleucine concentration begins to drop down, that's not going to be there anymore to be able to fit into the enzyme, and you're going to have more active enzyme now available, and threonine will then get catalyzed into intermediate A. Kind of neat. So there it is. Okay, there's the enzymes. This involves some practice. This involves some looking at it. We've got the lab, but it's all good. So for this, make sure that you can do these things. Explain how enzymes act as catalysts, what the induced model fit is, the role of enzyme-catalyzed reactions, the relationship between their structure and function, and the modes in which they are regulated in a system. So with that, I'm going to bid you guys good night, and we'll catch you guys in class later.